so diverse? And I hope you will help me to answer this question. I actually try to give you my view. So, as you probably all know, if you record the activity from the brain and you look at the responses of individual neurons, they're incredibly diverse. And you see that the neurons are responding to many different task-relevant variables, typically in a very complicated way, in a highly nonlinear way. And every neuron is pretty much unique in the kind of response that it has. So is there any profound computational reason for all this diversity? We believe that there is, and that's what I'm trying to convince you of during my presentation. So it will be supported by some experimental data, so I'll be talking about the real brain. But I will start with a long introduction about computational reasons why we should have this kind of a diversity. And then I'll switch to the experimental evidence. Okay, so let me start with some very general considerations. So let's say you're an engineer and you need to build a device that is specialized at performing a very simple task. So let's say, for instance, the device is observing a scene like this one here. This is a farewell. You see a couple, they're embracing, and they're probably saying goodbye on a platform before embarking on the train. And then maybe a locomotive will come, and then one of them will be boarding, and he or she will leave. Okay, so if you're an engineer, then probably what you would do is to design a device that is essentially looking at some of the features of these images, and it can recognize this particular situation as a farewell scene. So for instance, you may have two important features here, like a couple who is embracing, and then here the locomotive. And every time you see this kind of features, then you say this is a farewell scene. So if you want to design this device, you probably need to incorporate some elements that are selective to these two different features that are relevant for recognizing the scene. Okay, so this would be a very orderly machine, and then you can say this part of the machine, this particular device, is devoted at recognizing these two people, and this other one, for instance, is devoted at recognizing the locomotive. Now, if you look at the brain performing the same task, then what you would see is beautifully depicted by Umberto Boccioni, who is a painter from Italy, from the futuristic movement from the beginning of the last century. And, this is really a farewell. And it's part of a triptych that you can see in New York, which is called States of Mind, and I think it's not by chance. And you can see here that all the task-relevant variables are highly mixed. You see these embracing figures from different angles, probably at different times. You see the locomotive the front view, then you can see it laterally. Everything is completely mixed up. Okay. Now, I think that this is really what you see in the brain if you stick an electrode and you record the activity. And what I want to convince you of is that these representations are actually better than these highly specialized, engineered representations that you would use if you design one of these kind of devices. Okay, so let me start with the introduction that I promised. And I'm going to define every quantity because I think it's really fundamental that you get the ideas more than all the details about the experimental results. Okay, so let's consider the following situation. Now, you need to integrate just two variables. In real-world applications, of course, you need to do much more than that, but this is just to illustrate some of the basic concepts. So let's say that you are studying the responses of the neurons to a stimulus that is characterized by two parameters, A and B. So, for instance, they could be the parameters of a visual auditory stimulus. So there is a visual component, and let's say A is characterizing its luminance, and there is an auditory component, which could be sound intensity. Okay, this is just one example. Okay, so now here I'm going to define the response property of the neurons, and I'm going to say exactly what I mean by mixed selectivity, so when the neuron responds to multiple features or multiple task-relevant variables. But first I will start with the simple neurons, those neurons that I believe are probably in the brain by chance, and those are the neurons that many electrophysiologists are focusing on. Okay, so, here I have a very simple contour plot, and I have the activity, the firing rate in hertz of the neuron as a function of the two parameters. And you see that it increases along this axis, and it's constant along this axis. You will never see such a neuron, but again, I'm just defining the different response properties. So this is a neuron that cares about A, and it doesn't know anything about B. And analogously, you might have a neuron that cares about B, and doesn't know anything about A. Okay, so these are highly specialized neurons that have this kind of pure selectivity. So they selective only to one particular task variable. These are easily interpretable, and that's why very often people are looking for this kind of neurons in the brain, because then it's easy to attach them a label and say, this guy is an A neuron, this guy is a B neuron, and then maybe you can count how many of these neurons you have and so on. Okay, so another possibility is that you can describe the neuron as a simple linear function of the two parameters. Okay, that's already some mixing, but it's a simple form of mixing, and in particular, it's linear, so that's why I'm going to call it linear mixed selectivity neuron. Okay, so whatever is not described by these two simple models is what I'm going to define as a nonlinear mixed selectivity neuron. And here I have two examples. So for instance, in this case, the firing rate is a nonlinear function of A and B. This is a different nonlinear function. Typically, these are not easily interpretable response functions. Very often people discard this kind of neurons, but they contain as much information as the other neurons, at least in principle. And of course, in order to decode one particular variable, let's say A, it's not sufficient to look at a single neuron, but you need a population of neuron, and then you can determine the value of all the different parameters. Okay, so now that I define these two different categories of responses, what I want to show you is that neural representations based on these different types of responses have a very specific geometry, and I'm going to define that in a second. Then I will show you why we care about the geometry, because the geometry is really directly related to the ability of a readout to perform tasks. And I'll show you that when you use these pure selectivity neurons, there are a lot of things that you cannot do. Actually, the vast majority of the possible tasks that you can imagine. Okay, so let me start with a very simple example. Now I'm going to consider only pure selectivity neurons. So let's say F1, which is the first neuron, is just simple function of A. F2 is a simple function of B. F3 is a different function of A. And now I'm going to use this representation. I'm going to use this firing rate space, where along the coordinate axis, I have the firing rate of all the neurons that I'm considering. Okay, so this is a 3D space. If you have n neurons, you're going to have an n-dimensional space, and that's what you actually should consider when you are going to analyze the neural activity of the brain. But for now, just to illustrate the main concepts, let's consider a simple case that I can visualize. Okay, so now let's say you run an experiment. You present a stimulus, let's say stimulus A, that is characterized by A1 and B1. These are the values of the two parameters. And then you record the activity of these three neurons. So then you get a triplet of firing rate. That's a point in this space, or a vector if you prefer. And I'm going to represent it with a sphere so it's more visible. Okay, now you repeat the experiment, but now you present a different stimulus. Again, you look at the activity in one particular time being, let's say during the visual response of the neurons, and you get presumably a different point if you are in the right area, if you are in an area that is encoding these different stimuli. Okay, you can keep going on, and let's say, no, I stop after 4 stimuli. Okay, there is one important property that this set of points has, and the property is the following. that you can always find a plane that contains all these points, okay? It's pretty clear from the equations that now are not here any longer, but essentially you're always going to have that these neurons live on a low-dimensional manifold. In this case, it's just a plane. Okay? So what I mean by dimensionality here is the minimal number of coordinate axes that you need to specify all the points. And if you properly align two axes, one along here and one along there, then you actually don't need the third axis. Okay? So this is a low-dimensional neural representation. And I'm going to show you that this is bad. And, actually, this is well known in the machine learning community, but I want to summarize all the main results. Okay, so this doesn't depend on the number of points. If you increase the number of points, you repeat the experiments. If these neurons are, these firing rates are a linear function of the parameters, all these points will be contained by the plane. And also, if I replace one neuron with a linear mixed selectivity neuron, or actually I replace all of the three of them, then the plane is only going to be tilted, but it will have the same dimensionality, okay? So same geometry in a sense. Okay, so, before going to the beautiful world of high-dimensional representations, let me tell you why we care about the geometry of these neural representations. So I want to try to introduce a measure of the quality of the neural representation. Can I say whether a neural representation is good or bad? Okay, in order to do that, I need to come up with a task. So let's say I need to perform the following task. I have these 4 points that represent 4 different stimuli, and then I want to train a readout to respond to the stimulus that is represented by this point and not to the other 3. Okay? So this is a very simple task, and I can do it with a linear readout, which would be equivalent to finding a plane separating the red point from the yellow point. That's easy. Okay, let's consider a different task. So, most of you have already recognized this. This is equivalent to the famous exclusive OR problem, and it's known to be a non-linearly separable problem, which means that in this case, if I want to separate the red points from the yellow points, then I cannot do it with a linear readout. Okay, there is no plane separating the yellow point from the red point if these points are on a plane. I can do it with a more complex readout that would correspond to a curvilinear surface like this. Okay, this actually can be a very complicated task for the readout, and you're essentially moving the problem downstream. And actually, this can become an extremely difficult problem if you increase the number of points, because then the number of changes in the convexity of this curvilinear surface will increase with the number of points. So, what I'm trying to say is that you could rely on this kind of curvilinear surfaces, but it's not really a scalable solution. And it's actually much easier to use as an input high-dimensional neural representations. Okay, so as I promised, let's go now to nonlinear mixed selectivity neurons, and let's see what happens to the geometry of the neural representations. Okay, so here I have F1 and F2 as in the previous case, but the third neuron has now been replaced with a neuron that has nonlinear mixed selectivity. So F3 is some nonlinear function of A and B. And now you see that essentially the 4 vectors representing the responses to all the 4 stimuli span all possible dimensions. So they basically define a tetrahedron. They're not on a plane any longer. Okay, so this is 3D, and it's as high dimensional as it can be. With 4 points, you cannot do more than a tetrahedron. Okay, now the nice thing of this kind of representations, and this is the basic trick of support vector machines, is that even if you use an extremely simple readout, like a linear readout, which presumably can be implemented even by individual neurons, now you can separate them. So this would be a plane, and I don't know if this is clear. Maybe I should rotate it a bit. So then you can see that the yellow points are on one side and the red points are on the other side. Okay, so I generated some more complicated representations, And it's really fundamental that at least one of the neurons has this kind of nonlinear mixed selectivity. And in this case, I can solve any classification, any binary classification problem in this specific example. Okay, so let me summarize these initial considerations. I know that these are familiar to many of you, but I need to define everything to get them to the experiments. Okay, so if you look at the single neurons, you can have two categories: pure linear mixed selectivity, which would be low dimensional, and every time you have nonlinear mixed selectivity, you're going to have a high dimensional representation. Now, in this case, you need a complex readout, a curvilinear surface. In this case, with a simple readout, you can do any binary classification of the points. So you can do a lot of tasks. And it would be great to find a quantitative measure of the quality of the neural representations that is based on the complexity of the readout. That's very difficult. So we're going to use a different strategy. Instead of evaluating the complexity of the readout, we are going to focus on one particular simple readout. So let's say the linear readout, because that's something that we can control very easily, and then we can do calculations and so on. So for now, it's totally arbitrary. But I'll show you that in many experiments, it actually coincides or it's compatible with the readout that is used by the animal. So for now, it's just a readout that is not arbitrarily complex. So I need to limit the complexity of the readout, and I choose the linear one. And then I'm going to count how many colorings or how many binary classifications of the inputs I can implement with this linear readout. So the larger, the better, of course. Okay, so, in general, I'm going to have n neurons, which will be the number of axes. I cannot represent them, so this is, just a cartoon. And then p would be the number of points, or if you prefer, the number of spheres in this n-dimensional space. And I'm going to consider the cases in which p is smaller than n. So p will actually set the maximum dimensionality. I'll come back to this maximal dimensionality. Okay, so then you start with some coloring, and you see whether a linear readout can implement it. If it does, then you say, okay, this one counts as one implementable coloring. There will be colorings that are not implementable. And then, I go over all possible colorings, which will be 2 to p, so it can be very large. And NC would be the total number of binary classifications that are implementable by a simple linear readout. Okay, so all where I have this green mark. Okay, so now what you can realize by doing some calculations is that this number, the number of binary classifications implementable by a linear readout, depends exponentially on the dimensionality of the neural representation. So this is telling you two things. The first one is that the computational advantage of high-dimensional representations is huge because it grows exponentially. And the second one is that you can measure dimensionality, or you can characterize the geometry of the neural representations in two ways. One is to essentially count how many independent axes you need. The other one is simply by counting the number of binary classifications that are implementable by linear readout, because that's directly related to the dimensionality. And actually, that's what we're going to do, because it's much more robust to noise, as I will show you in a second. Okay, so this is, again, summarizing all the single neuron properties, dimensionality, and here, this NC will be our measure of the quality of the neural representation. Okay, so now there is a problem. So let's go back to the low-dimensional case. So as you remember, we cannot do this task because there is no plane separating the yellow points from the red points. Okay, you might say at this point that this is not entirely true, because as soon as you introduce noise, the probability that these points are exactly on a plane is 0. And you would be perfectly right. And given that in the brain you have a lot of noise, this is going to be a problem. So, this is essentially what is said in the title. A low-dimensional neural representation, if you introduce noise, may look high-dimensional. However, there is something that is preserved when you introduce noise, which is your inability to perform this particular classification task. Because now, if I have to rely on noise, I can find a plane, you know, let's say this one, that coincides with the original plane where the points were. Okay, so now the yellow plane is separating the yellow points from the red points. However, this is true only for this specific realization of noise. So if I train my classifier on these points, and then I test it on a different realization of noise, I'm going to fail miserably. Okay, so for instance, this will be a different one, and of course it will be on the wrong side of the plane. Okay, so you know, this is something trivial. I cannot rely on noise to do reliable classification. So you know, your inability to perform the task is something that is preserved when you introduce noise, and that's what we can use to estimate the geometrical properties of these representations. Okay, so we are going to measure dimensionality in the following way. We're going to count the number of binary classifications that are implementable by a linear readout. And what we mean by implementable is that the cross-validated performance of your linear readout should be larger than, let's say, than some threshold. Actually, the dimensionality, I mean, the final dimensionality will be proportional to this NC, and the proportionality factor will depend on theta. Okay, so this solves the problem of noise. However, now what can happen in the brain, and actually it does happen, is that you might have a situation like this, which would be fine. So this is a high-dimensional representation where you should be able to separate yellow points from red points, and this would be different realizations of noise. But you might have a situation where the noise is really too large. And since we are going to analyze the data on a trial-by-trial basis, this is actually what happens. Okay, so you know, here what can happen is that you have the right geometry, but you don't have enough neurons to actually train a decoder to perform this particular coloring. So what you need to do is to play this game only when n is large enough. So in principle, you need to compute this NC for a progressively larger number of neurons, and when you reach an asymptote, then you know that this limit is due to the geometry, and it's not due to the too much noise that you have in your data. Okay, so this is essentially the procedure. And now it's something that you can do in the real brain. Okay, so let's consider now prefrontal cortex, which is known to be really messy in terms of responses. Basically, every neuron is pretty much unique. There is almost no anatomical organization, at least on some spatial scale. And it's really the perfect candidate for estimating the dimensionality. Okay, so we used the data collected by R. Miller at MIT. This was the original paper that came out in 2010. And you know, we needed a task that is complex enough so we could have enough points in this n-dimensional space. And this was definitely one of the most complex tasks I've seen that they used to train a monkey on. Okay, so it's a memory task. And there is a sequence of two stimuli, so fixation point, and there is a first cue, a delay, second cue, then there is a second delay, and then the monkey has to look at this sequence of stimuli, remember it, and then there are two ways of testing the memory of the monkey. So there would be blocks of trials where there is this recognition task. I'm going to describe it in the next slide. And blocks of trials where there would be the recall task. The recognition task was like this. So this would be the sample sequence that I already showed you. And then there would be either a matching sequence or a non-matching sequence. And the monkey has to hold the bar at the beginning of the trial and release it only when it sees the matching sequence. After a non-matching sequence, there would be always a matching sequence. The second possibility is the recall task. So sample sequence, and then there would be three stimuli simultaneously on the screen, and the monkey had to saccade to the two stimuli of the sample sequence in the same temporal order. Okay, so let's look at the firing rate activity. Okay, this is a typical neuron, okay? We didn't select it in any way. It's really one of the typical neurons. And the only point I want to make here is that it's messy. It's very difficult to say what this neuron responds to. And that's why I believe it usually doesn't make much sense to try to attach labels to the neurons. Okay, so this is a classical PSTH. And you see each of these traces the firing rate of the neuron in a different condition. So the first Letter denotes the type of task, recognition or recall. The second one is the identity of the first queue. And then I just averaged over the second queue because otherwise it would be too crowded. And you see that the activity changes in time. The response properties change depending on the context, depending on all the other task-relevant variables. You're not going to have the same response to Q1 and Q2, so the selectivity is not an inherent property of the cell. This is true everywhere in the brain, including in sensory areas. It's just that it's usually ignored. Okay, so if I focus, for instance, on the activity here, you see that the firing rate of this particular neuron in these different conditions is strongest to this particular combination of stimulus C and task type 1. Okay? So this neuron really likes stimulus C, but mostly when it's in task type 1. And this kind of profile is different for every neuron, and it's different for different time epochs, even if you consider the same neuron. Okay, so this is really a signature of these high-dimensional representations. But we want to prove that this is not just noise. It's something that you really can use, that monkey can use. Okay, so let's focus on this time interval. Okay, this is published. If you're interested, you can see what happens in these other time intervals in the paper. But I just want to make the main point here. So here, it's where presumably the monkey has all the information it needs. Okay, and I want to put some numbers here. So, we had 4 visual objects for Q1. Same 4 visual objects also for Q2, but Q2 could not be the same as Q1. Otherwise, this would be easy trials where the first Q and the second Q are identical. So only 3 possibilities for Q2, and then 2 different task types. So the total number of conditions is 24, which is the total number of points in this n-dimensional space. And this is also the maximal dimensionality, because we have many more neurons. So if you have 2 points in an n-dimensional space, there is always a line going through them. So the maximal dimensionality is actually 1 plus the offset, because the line maybe doesn't go through the origin. So it's equal to the number of points. It doesn't matter how many axes you have. Okay, so before going to the data, to the real data, I want to do a simple exercise. And I want to say, let's see what happens if I use only pure selectivity neurons in an ideal case. Okay, so I want to show you that these are bad representations. And this, if you find these representations, you're probably not in the right brain area. Okay, so let's consider a population of N neurons that encode task type perfectly. They really, your ideal neurons where you can attach a label and say, this is a task type neuron, but they know only about task type. They don't know anything about Q1 or Q2. Okay, so then you have recognition task, and you would have a point in this n-dimensional space. Again, this is just a cartoon. And then another point for the recall task. So the maximal dimensionality would be 1D, as I already told you. Okay, for Q1, so I have the second population here. This is a segregated population, and it's highly specialized at encoding Q1. So in this case, I have 4 points. The maximal dimensionality is 3D. And the same for Q2. Okay, so now let me consider a population of neurons that comprises all these 3 different populations. So here I have all the information I need about task type, Q1, and Q2, but it's represented in this format where I have only highly specialized neurons. Okay, so in this case, the maximal dimensionality is 1 plus 3 plus 3 plus the offset, as I mentioned before. So it's 8. Whereas the total number of combinations, as I told you, is 24, which is potentially the maximal dimensionality. So there is a huge discrepancy in terms of number of binary classifications you can perform when you use these highly specialized neurons, or when you consider the more general case where you have also nonlinear mixed selectivity. Now, it's 2 to 8 versus 2 to 4, basically. So it's a huge difference. Okay, so now in our specific case, we had 237 neurons. Not all the neurons were simultaneously recorded, but we pulled them together. And 24 points. So we had 17 million possible binary classifications, or if you prefer, colorings of the points. We went over all of them. Actually, you don't need to do it, but as a control, we did it. And then you can use some of the trials to train your linear decoder and some other trials to test it. Everything is on a trial-by-trial basis. Okay, so you have a lot of noise. Okay, and these are the results. So here on the y-axis, I have the number of binary classifications that are implementable by this linear readout. Okay, it's cross-validated performance. It's on a logarithmic scale as a function of the number of neurons, because as I mentioned, I need to increase progressively the number of neurons and see whether I reach an asymptote or not. Okay, so this is some fake data. It's synthetic data where we have only pure selectivity, but with the same noise that we have in the real data. And this is the real data. And it goes all the way up to the maximal dimensionality. So this will correspond to the number of dimensions. So it will be 24. This means that you can perform all possible 17 million tasks on these 24 points, which is probably more than what you need, but it gives you a huge flexibility, which is certainly needed in a brain area like prefrontal cortex. Now, you might have noticed that here the number of neurons goes up to 4,000. So the problem is that if you stop at 237, which is about here, you don't reach the asymptote. So we had to resample the neurons. We were very careful at resampling the neurons without increasing the dimensionality, but we essentially had to reduce the noise in order to reach the asymptote. Actually, all the effects I'm going to describe, including this one, would be true even if you don't resample the neurons. But we wanted to get a number, so that's why we went all the way up to 4,000 neurons. Okay, so, the dimensionality seems to be maximal. This is really a usable dimensionality because it can be used by a linear readout. Okay, so it's not that I'm counting dimensions that just correspond to noise or to global fluctuation of the activity. This is all cross-validated performance. So it would be nice to know whether these high-dimensional representations are actually used by the monkey. So, ideally what you would like to do is to shut down all the neurons with nonlinear mixed selectivity. And it would be great to do that kind of experiments, but it's only becoming possible now and only under very specific conditions. So, at that time we couldn't do it, but we did something else, and we were lucky enough that the monkey was making enough errors that we could analyze the errors. So the idea is that if for any reason the dimensionality collapses, okay, typically it's not a reason that is under control, then we should be able to see that the monkey is going to make a mistake. It will still be a correlation, but it will be a strong indication that probably the dimensionality is used by the animal. Okay, so, when I said we had enough errors, I actually was referring only to one of the two tasks, the recall task, because for the recognition task, the monkey was too good. So we had to restrict our analysis to only this task, which means that now we have 4 by 3 different conditions, so 12 different conditions in total, which means 12 points, which means 12 maximum dimensions. But still, it's wide range where the dimensionality can vary. Okay, so this is what you get. So here on the y-axis, you have again the number of implementable classifications, and here the number of neurons. And the black line is for correct trials, and the gray line is for the error trials. Okay, so you see there is clearly a collapse in the dimensionality that is predictive of behavior of the monkey. Now, there could be a lot of trivial reasons why the dimensionality collapses. Okay, so let's say the monkey didn't see one stimulus in that particular trial. Then, of course, the dimensionality will collapse because the linear readout will not have the information about that particular stimulus. So we were lucky because it didn't have to be like this, but actually there was no trivial reason for the errors of the monkey. And in particular, if you try to decode again on a trial-by-trial basis, the identity of the two stimuli individually. Okay, so you focus first on Q1, you try to decode it in many different epochs of the trial, and then you focus on Q2. You could do it with a very high accuracy, both in the correct trials and in the error trials. Okay, so this is represented here. The green line is the decoding accuracy for Q1, and the solid line is for correct trials, and the dashed line is for the error trials. You see, it goes all the way up to 100%. You can tell for sure what is the stimulus that the monkey has seen. And you can do the same also in the error trials. So it means that the information about the cues is there, but it's not in the right format. It's probably in this low-dimensional format. That's why we see this collapse in the dimensionality. Okay, so this is very subtle, and it took us a while to understand what's going on. So I'll try to explain. But the point I want to make here is that we got 19 pages of comments from the reviewers. They wanted all sorts of controls. And we ran all of them, and basically the dimensionality was the only quantity predicting the behavior of the monkey. You can see the details in the 58 pages of supplementary information. Okay, so let's try to understand what's going on. Okay, let's go back to individual neurons. So for each neuron, what you can do is to try to fit a very simple linear model to the response of the neuron. Okay, my goal is to separate the pure selectivity neurons from the nonlinear mixed selectivity neurons and see how much they contribute to the dimensionality and what happens in the error trials. Now, what we realized immediately is that it doesn't make much sense to divide neurons in two categories because every neuron has a linear component and a nonlinear component. Some of the neurons have a stronger linear component, some others have a weaker one. So what you can do is to characterize these two components for every neuron. So this would be essentially the linear model. So basically this psi is a binary variable. It's 1 when the condition in the parentheses is met, 0 otherwise. So it's what you would do in ANOVA, in many classical types of analysis. So, and this would be the coefficients. But it's just that, this would be the linear component. And these can account for at most 6 dimensions. Okay? You can count these dimensions. Basically, it's the number of variables that you have, the number of independent variables that you have. Okay, so this can account for 6 dimensions at most. And you remember the maximum dimensionality is 12. Now, whatever we cannot explain with the linear model, so the residuals of the linear model is basically the nonlinear component. And that accounts for another 6 dimensions. Okay, there's one important point I want to make here. When you play this game, the percentage of variance that is explained by the linear component is about 85. Okay, so most of the people would stop there and they would say, I have a good model. It explains 85% of the variance. But actually, as I will show you, this 85% of the variance is not correlated with behavior. You really need to look at the nonlinear component to be able to predict what the monkey is going to do. Okay, so let's say you do the analysis only with the linear component. So it's again the dimensional analysis, number of binary classifications as a function of the number of neurons. And you see that these two curves are practically identical for correct and error trials. So this means that the linear component doesn't change at all. Basically, the pure selectivity neurons that you find, and there are many that you can find by chance, or those that have a strong pure selectivity component, it doesn't change. Whereas the nonlinear component that is really fundamental to expanded dimensionality is also very fragile to noise. And then actually what is essentially showed by this plot, where you see that these two curves, correct and error, actually diverge from the very beginning. So the collapse in the dimensionality is entirely explained by the nonlinear component. So it means that, first of all, you really need to look at these small components. If you do PCA, you need to go to the 7th principal component to see any difference between correct and incorrect trials. And these small components, these very difficult to interpret small components, are actually fundamental in this task in order to generate the correct response. Okay, so, I'm almost at the end, but I still have about 10 minutes. And I want to summarize a bit some of the recent unpublished results, we're still working on them, that we obtained in different brain areas. Because we believe that this is a very important and general principle, so it's probably true almost everywhere. Okay, so this is one example. We did the same analysis also in OFC, in ACC, even in the amygdala, where you had a lot of structure. We see basically the same results. Okay, so this is a freely moving mouse, and we're using the Schnitzer miniscope. So it's really nice because it's a commercial product, you can buy it, and then this is the kind of neurons that can be detected by the standard software. And you can do calcium imaging, and then you get all these traces for these neurons. We started from the dentate gyrus because, first of all, because there is not much about the dentate gyrus, because the activity is so sparse that there are not many electrophysiological results. So it's interesting to look at this brain area. But also because Lloyd Gebet and colleagues in the Moser lab, they know that there is a lot of diversity in their responses, probably more than in CA3 and in CA1. And we actually believe that the dentate gyrus is a sort of an intermediate stage for expanding the dimensionality before passing the information to CA3 and CA1. Okay, so what I'm going to show you is essentially something about the properties of the individual neurons. And I don't have time to go over all the details, but let me show you, first of all, that you can do something with these calcium traces, because it's not obvious a priori. And so let me run this movie. Okay, so these are the calcium traces here. Okay, you don't see much. I mean, from time to time you have these big events. We're actually not sure that these big events are encoding much of the information. We certainly know that even these wiggles here encode a lot of information. Okay, so we don't know how to interpret this activity. There are still many issues when you do calcium imaging, but we don't care because we just go directly and we decode the position. Again, everything is cross-validated, so we use some data set for training, some other for testing. Okay, so this is a video of the animal seen from above, and we overlaid essentially the decoding accuracy, the decoded position that you can obtain from a very simple linear readout. Now, if we are right and these are high-dimensional representations, a linear readout would already do an excellent job. And actually, that's what happens. So let me run it again. So the blue line is the actual trajectory of the mouse. This is a 10-pair video, okay? So it's much faster than what it actually is. And the red one is the decoded position. So now you see that every time the animal stops, that's well known, you lose the position. It goes all over the places in the arena. But when the animal moves, it's actually pretty accurate, given that, again, this is calcium traces and this is the dentate gyrus. So its activity is much sparser than in many other brain areas and also other parts of the hippocampus. So, we typically get something like 10 centimeters resolution, which is pretty good, even compared to CA1 and CA3. Okay, this is just to tell you that this is not just noise. Okay, so if we compute the dimensionality of this, probably doesn't make much sense if it's just noise, but it's something that we can actually use to do some kind of decoding. Okay, let's look at individual cells. What you can do is to come up with a criterion to classify the cells. We use the same criteria as Lloyd et al. And then you can identify what people normally call place cells. And again, these are easily interpretable cells because you basically have that the cell responds only when the animal is at a particular location. Okay, we already seen this kind of things the first talk in the morning. So you're already primed. So each of these boxes is a different cell, and basically this represents the arena. And then the activity of the neuron is color-coded. So here it's highest when the animal is in this corner, and it's lowest when it's here. Okay, these are all well-behaving cells, and actually these are the top 20 place cells that contribute the most to the decoder. Okay, so you can take the linear decoder, look at the weights, and then from there you can determine what are the important neurons and what are the non-important neurons. Okay, place cells that are classified using this criterion are about 25-30% of the cells, depending on the animal. And the other cells, what we call the non-place cells, of course, some of them could be considered also place cells with multiple fields, but only to some extent. They're much more difficult to interpret. Some of them really look like noise. They're all normalized in the same way. So here, you just see these kind of shades. And still, these are the top 20 cells for the decoder. If I remove them, I will be really in trouble to decode position. Okay, so now you see all these non-place cells. These are the top 20. Okay? And now you can play the following game. So are these neurons that have been studied so much really the most important and valuable neurons when you want to determine the position using a decoder? Okay? Or do these other weird guys also contribute to the position, to encoding the position? Okay, so then you can take the top 100 cells, the top 100 place cells, the top 100 non-place cells. We had, on average, between 300 and 400 cells. And look at the decoding accuracy. Okay, so here I'm plotting the error in centimeters. So this is the difference between the actual position and the decoded position. Mm-hmm. I'm considering all time beings, also when the animal doesn't move. Okay, so that's why it's slightly higher than what it could be, and it cannot be compared directly to other studies. But you should consider this as an upper bound of the error. Okay, so now if I just use a decoder that is entirely based on the prior distribution. Okay, so visitation is not uniform. The animals tend to spend more time near the walls, not in the center. So if I use the prior, then I can already do a pretty good job at guessing the position of the animal. Then I get an error that is about 12, 13 centimeters. Okay, so now if I use the place cells, this error goes down significantly. Actually, we know from a different type of analysis that it's significantly different from chance. It's really a pretty good accuracy, especially if you just restrict yourself only to the time beings when the animal is moving at a certain speed. This unfortunately contains everything because it's something that I got yesterday night, and so we're still working on it. So actually, this would be much lower. But this is for the place cells. And now you see that the weird guys give as much contribution as the place cells. Actually, if anything, they do slightly better than the non-place cells, than the place cells. Okay, this would be the performance when you consider all of that. Okay, so this is just to say that, again, you don't want to discard this kind of neurons. These are important actors, and they can be actually extremely important for any kind of computation that you want to consider. And so this prepares me for my conclusions. So the neural representations in prefrontal cortex, they're high dimensional. Okay, so this is a combination of two ingredients. You really need both of them. One of the two in isolation is not sufficient. You need nonlinear mixed selectivity, and you need diversity. Okay, so you need some nonlinearity in the function of the firing rate as a function of the task-relevant variables, but you also need sufficient diversity. So you need many different combinations, many different nonlinear functions of these variables. Okay, dimensionality is correlated with behavior. So this is interesting in itself, but it also tells you that the choice of the linear readout, which initially was a kind of arbitrary, it's not totally unreasonable, because at least it predicts the behavior of the monkey. So the linear readout is compatible with the readout that is actually used by the monkey. Okay? So it could be coincidental, but it's not a trivial result, because, for instance, if you use a nonlinear readout, we know that it will be able to perform the task also in the error trials, because the information is there. It's just not in the right format for the linear readout. Okay, let me thank all the people who were involved in the projects that I presented. So Mattia Rigotti is really the protagonist, and he did most of the analysis. He was at Columbia, is now an independent researcher at IBM. And Omri Barak was also a postdoc at the same time. He's now a professor at Technion. And the data was coming from MIT, collected by Melissa Warden and Nerv Miller. And we had some collaboration with Xiaojin Wang and Nathaniel Doe at NYU. And for the hippocampal data, all the data was collected in the laboratory of René Hen at Columbia by Mason Kerbeck, and the analysis was performed by Fabio Stefanini and Ludmila Kushner. Okay, I will stop with the second painting of Umberto Boccioni. This is States of Mind 2, those who go, so basically the people on the train. If you want to see States of Mind 3, you have to come to New York, because I don't have it. And I will stop here. Thank you for your attention.
Thanks a lot for the talk. Questions? I would start asking you whether the nonlinear selective cells, since they are apparently more sensitive to noise, or their performance, the overall performance are more sensitive to noise, whether they have some difference predicted by physical feature that one might hope to find, or whether this is a result of the synaptic architecture, and so there is no chance to see differences.
Okay, so we don't know, but what we know is that if you take just random connectivity, that's an extremely surprisingly good way of expanding the dimensionality. So that would be sufficient in most of the cases. Now, we tried to fit a model with random connectivity to this data. I didn't have time to show it. And the model does a decent job, but if you introduce any form of learning, you do significantly better, indicating that you probably need the random connectivity as a substrate to generate all this diversity. But then on top of that, there is some degree of specialization that you acquire with learning. And that's certainly something that helps you to generalize. So if you want to generalize to many possible situations, you need to trade the dimensionality for generalization. And that's something that you probably can do by introducing some structure in the connectivity. Now, the diversity can be generated in many ways. And I mean, the morphology could certainly play a role, but we just don't know.
To the last graph, where you show how much explanation is there in the place cells and the non-place cells and all of them. Non-place cells and all cells are kind of equal. I mean, I'm surprised. I was expecting that you were going to gain in predictivity when you use all the cells. And I have then two questions. One is, how much better can we do with maybe state-of-the-art deep learning, like if we actually, in view of the discussion we had yesterday, and what are all the neurons then encoding for? If
okay,
not
so
for complete understanding of space.
Yeah, so I didn't have time, but if you plot the performance of the decoder as a function of the number of neurons, then you see that even in the cases in which you have 500, 700 neurons, the top 100 neurons are actually sufficient to give you the best performance, which is related to the fact that actually the dimensionality of these neural representations is limited by the size of the arena and the resolution by which you can represent space. So if the arena, in our case, is 50 by 50 centimeters, and you have a resolution of 10 centimeters, the number of independent points you can have, roughly speaking, is 25. It's 5 by 5. That sets an upper bound on the dimensionality. So it means that instead of counting the neurons, but it's related to that, you just do PCA, 25 components would be sufficient to give you the maximal resolution that you can achieve here. That's why if you consider 100 cells, and they're really the top 100 cells, it doesn't matter too much whether it's placed, non-placed, or whether you consider all the cells, because you don't gain much by introducing these additional cells. You already reach the asymptotic performance with a smaller number of cells. But yeah, we estimated the dimensionality, and it's as high as it can be in this situation. So the dimensionality is maximal also in this case, but we cannot do the cross-validation that we did in the other case.
What's the precision that the mouse has in its own head about the position it occupies?
We don't know. It would be interesting. It's something we were wondering. I suspect it's better than 10 centimeters, but many people believe that it's not much better than the size of the animal. So we don't know. To be honest, I really don't know in the case of the mouse.
I have another brief question. I'm fascinated by the fact that by feeding into the decoding algorithm, the raw calcium signal, you actually are able to reconstruct the position. So you don't need any pre-processing of the calcium signal to extract the time of presumed action potentials. Could it be therefore that you are indirectly and with a lot of noise accessing somehow the equivalent of subthreshold voltage or the correlate of subthreshold voltage fluctuations or synaptic inputs? So not yet superthreshold activity, everybody else would discard the data and would only look at sharp transient. Are you looking at noise or still might be action potentials?
Okay, so we don't know if it's action potentials. We don't know what it is, whether it's subthreshold activity or not. We know for sure it's not noise because we can decode position in a cross-validated way. So even if we eliminate all these big events that usually people are focusing on, we can still decode the position. So we don't know what it is, but I think if you do any kind of pre-processing, you risk to throw away a lot of information. So we decided to keep everything. Actually, what we are going to do is to not even consider the calcium traces, but directly the image. So before any kind of pre-processing. Of course, there are artifacts due to the movement of the animals and so on, but we believe that that's probably the right approach. If you just want to do decoding, you should keep all the information you have. Then if you're interested in the interpretation, then you need to make another step and try to make hypotheses about this activity. But yeah, I would certainly recommend not to throw away whatever is not identified by the software as a spike.
And another recommendation that you would have is to do simultaneous recording. So is the era of single electrode, single unit recording over?
Well, not necessarily. I mean, in the case of the prefrontal cortex, we had 237 neurons, and we had at most 15 neurons that were simultaneously recorded. Now, we just combined all these neurons together, so most of them were not simultaneously recorded. Now, you might wonder what happens when you introduce the temporal correlations that we discarded. So we played that game. And in that case, I mean, we were very limited by the fact that the number of simultaneously recorded neurons was small, but essentially the decoding accuracy doesn't change at all. So, it could be that the decoding accuracy goes down, it goes up, remains the same. In our case, it remained the same, both here and in the data of Daniel Salzman that I didn't have time to show, where the number of neurons that were simultaneously recorded was up to 70, so a much larger number. So in the data that we considered, there is not much difference. Of course, it could be different in different tasks or in different brain areas, but so far the temporal correlations didn't play an important role. In order to rule out that they actually play a role, you need simultaneous recording.
So I was wondering if you have any idea what happens when you consider only neurons that have shared variability. So neurons that's in different, in the same trial they vary together, and neurons that don't vary together. Do you think that plays a role?
Okay, I guess it depends on the time scale that you're considering. When you're saying varying together, this can be interpreted in two ways. One is if you look at the temporal correlations on the millisecond scale. Okay, or it could be that you look at the firing rate in one particular time being. And then, of course, the decoder is relying on these correlations because, I didn't have time to show it, but you can take these neurons, eliminate what people would normally call the traditional selectivity, let's say task type, for instance. I look at all conditions in which I have task type 1, all conditions in which I have task type 2, recognition or recall. And then we have a procedure to make this neuron essentially insensitive in the sense that the average, and actually all the moments of the distributions are the same in response to task type 1 or task type 2. And still, you can decode task type because you can rely on the fine structure of this complicated profile when you look at all the different conditions. So these are certainly correlations you rely on in order to extract information. I'm not sure it's what you had in mind, but... Yeah, again, I'm not sure I understand what you mean by decorrelated. We can discuss later, maybe.
Yeah, hello. Fantastic talk, amazing work.
Thank you.
But maybe a very naive question. I was wondering about place cells that you record in the dentate gyrus. Yeah, as I said, maybe it's a very naive question, but is that already described, or do you need to elaborate on that, on the place cells in the dentate gyrus?
You mean whether in the literature people already observe?
Because to my knowledge, they are
in the
not
paper
well
by
described.
Leutgeb and the
So I don't know if you
Mosers.
need to elaborate or not.
No, people see exactly the same kind of fractional place cells that we observe by using traditional electrophysiology. There are not many papers. I mean, the only one I'm aware of where they do this kind of analysis and they count the number of place cells is the paper by Leutgeb et al. But they do this analysis and they count the number of place cells, so they know the place cells are there.
And then you get response from all of them, or
sorry, say that
you
again?
get response from all place cells in the dentate gyrus. Not that you have just a small percentage of response, but you get it from all of them.
I mean, we have a few hundreds of cells.
Yeah,
The dentate gyrus contains about 1 million cells, so it's that's what we can do. I mean, I'm not saying the place cells are useless, as I'm not saying that the linear component is useless, because that accounts for half of the dimensions you needed. But all I'm saying is that we should care also about the nonlinear component and also about the weird guys, the neurons that actually encode the information in a non-easily interpretable way.
Thank you.
You're welcome.
I'm just curious about the magic number of 100 neurons as a classifier which is enough to classify. Actually, like DiCarlo's study he also claims like 100 neuron magical number. I'm just curious, the upper dimension limit which you set, like 24, 25, is it consistent across different behavioral tasks?
No, it's not. It actually depends on the task.
Okay.
So that's something fundamental that now, Surya Ganguly has formalized in a very nice way. And you should look at this recent paper. So that's something you should always keep in mind when you design an experiment, because the maximal dimensionality is set by the total number of independent conditions that you have. So in our case, we had 24 possible conditions. That's the maximal dimensionality. That's a big limitation, because it means that if you have a too simple experiment, it doesn't make much sense to record from millions of neurons. So now that, it's becoming possible to record the activity of the entire brain, you should also choose an animal that has a behavior that is rich enough that you can do with something with all this activity. So, the maximal dimensionality is set by the task. It's also set by some properties of the neural activity, but the task sets an upper bound that, cannot be surpassed. So, of course, it depends on the complexity of the task.
I propose we continue the discussion afterwards during the coffee break, and let me thank the speaker again.